everyone. So today we are here for the live session week 6. I'm myself Sonam Pahuja and I will be taking your session for today, that is for cell biology, cellular organization, division and cellular processes. So for the week 6 we will be talking about chromatin organization, SMC proteins, chromosome organization, and further their real-time imaging of DNA loop extrusion by SMC complex, the cohesion complex and its function, the mysterious biological function of chromosome loops. So starting from the question number 1, which one of the following is not a feature of euchromatin? Loosely packed and open chromatin, transcriptionally active DNA. Is the screen visible?
Yes.
Okay. So which one of the following is not a feature of euchromatin? Is it loosely packed and open chromatin, transcriptionally active chromatin, highly sensitive to DNA is one, no association with HMG14 and HMG17? Anyone knows the answer? Anyone knows the answer? What is euchromatin? What is euchromatin? I hope everyone knows what is chromatin first of all. What is chromatin? Chromatin is composed of
DNA and protein.
yes, correct. DNA and histone proteins. And types of chromatin?
Heterochromatin and euchromatin.
Correct. So what is euchromatin?
Euchromatin is a loosely packed form of chromatin that is more transcriptionally active.
Yes, correct. And what about heterochromatin? What about heterochromatin?
tightly packed DNA.
What is its role? What is the role of heterochromatin? Okay, let me explain. Chromatin is composed of DNA, RNA, and histone proteins. And two types of chromatin are euchromatin and heterochromatin. In, in euchromatin, the chromatin fibers are loosely packed and they undergo the process of condensation and decondensation in the cell cycle, and they often remain active in transcription. Okay, since RNA is synthesized through DNA, so it is transcriptionally active. And due to less compact structure, they show light staining. They try to replicate in the early S phase. Okay, and they are more easily transcribed than heterochromatin, and they appear as beads on a string under an electron microscope. Now coming to heterochromatin, it is condensed form of chromatin. It is transcriptionally silent and highly condensed and tightly packed. It is formed by modifications to histone and recruit the silencing complexes. Okay. So chromatin plays role in DNA replication, transcription, repair, and cell division. And when cells divide, chromatin condenses to form chromosomes. Okay. And structure of chromatin is influenced by nucleosome position, histones, and other DNA binding proteins. So it is saying that which one of the following is not a feature of euchromatin. So it is loosely packed and opened chromatin. It is correct. It is transcriptionally active. It is also correct. Highly sensitive to DNA is one. You know what is DNA is one? What is DNA is one? DNA is one. It is an enzyme which degrades the DNA. Okay. So euchromatin is highly sensitive to DNA is one. Because it's relatively open and accessible structure, it allows the enzyme to easily cleave the DNA within it. So it, as it is actively transcribed, so if you talk about heterochromatin, it is less sensitive to DNA's digestion, while euchromatin, which is openly available and loosely packed, so and also actively transcribed, so it is more sensitive to DNA's one. Okay. Now talking about association with HMG 14 and 17. What is it? HMG, heard of HMG? People who are typing in the chat box, please, you can speak up, as I won't be able to view your messages while my screen is sharing. Okay. So HMG 14 and 17, these are chromosomal proteins. Okay, these are basically recombinant human chromosomal proteins. Okay, and these are highly similar in sequence and function, and often they act in regulating chromatin structure and transcription. And their role is mainly because of their ability to bind to euchromatin, and their role in regulating the gene expression, as they can enhance the transcription by facilitating the recruitment of transcription factor. They can help in recruitment of transcription factor to the promoter region of a particular gene. So transcriptionally regulation of these chromosomal proteins are tightly linked to the euchromatin. So it is saying that no association with HMG 14 and 17. So this is incorrect. Okay, as both of these proteins have a specific DNA binding domain that allows them to bind to the linker DNA between nucleosomes, particularly with this euchromatic region where the chromatin is more open and accessible. And by binding to nucleosomes, these particular proteins can influence chromatin structure also. It can cause slight bends or changes in fiber organization, which can make DNA more accessible for transcriptional machinery. Okay, so these particular proteins are associated with euchromatin, which is loosely packed and actively transcribed. They bind to the inner face of nucleosome within euchromatic region, facilitating access for transcription factors and RNA polymerase, thereby promoting the gene expression. So it is loosely packed and open chromatin, it is correct. It is transcriptionally active, it is also correct. It is highly sensitive to DNase 1, it is also correct. And not a feature of euchromatin, it is that it has an association with HMG 14 and 17. So this is an incorrect statement. Okay. Moving on to question number 2. Which one of the following is a feature of heterochromatin?
Loosely packed.
Is a feature of heterochromatin. You have to read the question very carefully. Earlier it was saying not a feature of euchromatin. Now it is asking it is a feature of heterochromatin.
Option D.
Option D, correct. No association with HMG 14 and 17. Because all these three options are correct for euchromatin. Okay. They are loosely packed, they are transcriptionally active, highly sensitive to DNase 1. And euchromatin is tightly packed, transcriptionally inactive, and also not very sensitive to DNase 1. And it also has no association with HMG 14 and 17, as they are linked to the nucleosome and linker DNA of euchromatin. Okay, so option D. Moving to question number 3. Transcriptionally active chromatin tends to be deficient in transcriptionally active chromatin tends to be deficient in ट्रांसक्रिप्शनली एक्टिव क्रोमेटिन कौन सा होता है, यूक्रोमेटिन या हेट्रो? Which is transcriptionally active chromatin?
Euchromatin.
So it will have HMG 14 and 17, earlier discussed. Now tell about H1. Will it have H1?
Option D.
Option D, chromatin remodelers. Okay. What is H1?
Linker DNA.
Yes. So euchromatin will definitely have that histone H1, linker DNA, right? And we already discussed about HMG 14 and 17. So chromatin remodelers, transcriptionally active chromatin, that is euchromatin, it lacks chromatin remodelers. Okay. So it contains histone H1 but is generally considered to be less densely packed with H1 compared to heterochromatin. Okay. Meaning H1 is more prevalent in heterochromatin where it contributes to tighter chromatin compaction. Therefore, euchromatin does have H1, it has a lower concentration of it compared to heterochromatin. So H1 is present in both heterochromatin as well as euchromatin. But the question is asking it tends to be deficient in. Okay. So euchromatin will not have chromatin remodelers. What is chromatin remodeler? As you all know that heterochromatin is the highly condensed form of chromatin. Genes are usually not expressed there as it is transcriptionally inactive. Therefore, it requires chromatin remodeling to become accessible for transcription. Okay. And chromatin remodelers are protein complexes that can modify chromatin structure to regulate gene expression. Also, euchromatin, as it is transcriptionally active, it also requires chromatin remodelers because it also requires the active gene expression. So without chromatin remodelers, gene expression will not take place. So euchromatin actually, like transcriptionally active chromatin, it requires chromatin remodelers to access its DNA for active transcription. Okay. So transcriptionally active chromatin also contains that chromatin remodelers. Okay. So HMG 14 भी है 17 भी है chromatin remodelers भी है. Now coming back to H1. H1 हमने अभी देखा था कि H1 है, हेट्रो में भी है और यू में भी है. बट इज इट रियली ट्रू कि दोनों में है? हेट्रो में तो डेफिनेटली है. यूक्रोमेटिन में इट इज जनरली लेस डेंसली पैक्ड. ओके. हेट्रोक्रोमेटिन में ही H1 का एक्चुअल में रोल आता है because euchromatin is less condensed, so euchromatin has a more open structure with less H1, and heterochromatin, which is more densely packed, and due to its high H1 content, it is tightly condensed and usually transcriptionally inactive. So when we talk about H1, it is basically in heterochromatin, and only euchromatin will have less of H1. Okay, so this question is although incorrect tends to be deficient in. So it's saying deficient. It's not it lacks. Okay, so is deficient in H1 as it is less densely packed with H1 if compared with the transcriptionally inactive chromatin, that is heterochromatin. Okay, so euchromatin does have H1 but it has a lower concentration of it as compared to the heterochromatin. Clear to all? So the answer to it is A, H1, and not the chromatin remodelers, as it also requires chromatin remodelers for activating the transcription and further gene expression. Clear to all? Is it clear? Should I move ahead? Is it clear to all? Should I move ahead?
Yes, ma'am.
Okay. Which of the following features of heterochromatin is not correct?
Option D.
What? Can you please repeat? Yes, option D. Is the screen visible?
Yes, ma'am.
Okay. So the here the answer is option D. Heterochromatin. Features of heterochromatin which is not correct. So first of all we will find which is correct. Okay. Heterochromatin is highly condensed chromatin, it is correct. Transcriptionally inactive, it is incorrect. Resistant to DNase 1, it is also correct, as we have studied that euchromatin is loosely packed, transcriptionally active and highly sensitive to DNase 1, and euchromatin has an association with HMG 14 and 17, while heterochromatin does not have association with HMG 14 and 17. So the incorrect statement is association with HMG 14 and 17. Which of the following processes affect gene regulation by altering chromatin structure? Modification of histone proteins, mRNA processing, chromatin remodeling, DNA methylation. And then options are as follows. Choose the one. Chromatin structure is affected by which of these following? Anyone knows the answer?
C.
C. 1, 3, and 4. Yes, correct answer. Can you explain? Can you explain the answer? Why is it correct? Or is it a guess? Okay, let me explain then. So it is saying which of the following processes affect gene regulation by altering the chromatin structure. So chromatin structure is affected by histone modifications, that is modification of histone proteins. Then DNA methylation is also one of part, and chromatin remodeling. So all these processes affect the chromatin structure and finally affects the gene regulation. Okay, so mRNA processing is incorrect. So all of these impact the gene regulation. These processes are epigenetic modifications or tags that are passed down through cell division. Okay, so here 1, 3, and 4 are correct. It is option C. Okay. So histone modifications, as we know that histone proteins are modified by adding or removing post-translational labels on the histone tails. For example, acetylation of histone tails is associated with transcriptionally active chromatin. Okay. Now DNA methylation, it is covalent addition of a methyl group to cytosines in DNA. Okay. DNA methylation is a dynamic process that increases with age. Now coming to chromatin remodeling, it is a process that opens the complex of DNA which is tightly wound into chromatin, and this also allows specific genes to be expressed. Okay, so all these processes alter the chromatin structure and finally affects the gene regulation, whereas mRNA processing is not a part of it. Moving on to question number 6. During eukaryotic transcription, chromatin structure is altered by the following modification of histone proteins. Is it acetylation, phosphorylation, methylation, or all of the above? Anyone? Anyone knows the answer? Should I explain then? Chromatin structure is altered by which of the following modification of histone protein? Is it acetylation, phosphorylation, methylation, or all of the above? In the previous question, I explained that in the modification of histone proteins, it could be acetylated. And also in DNA methylation, I already told that methylation of DNA occurs. So दो ऑप्शन तो यहीं ठीक हो गए, एसिटाइलेशन एंड मिथाइलेशन. एंड नाउ फॉस्फोरिलेशन. Phosphorylation is related to the chromatin remodeling. Okay, so here acetylation, here phosphorylation, and here methylation. So it is another form of this question. Okay, so here the answer is all of the above. Acetylation, phosphorylation, and methylation, all these alter the chromatin structure during eukaryotic transcription, with acetylation being the most widely associated with open chromatin and increased transcriptional access, क्योंकि हम यहाँ पर यूकैरियोटिक ट्रांसक्रिप्शन की बात कर रहे हैं. ओके, सो टॉकिंग अबाउट एसिटाइलेशन, दिस मॉडिफिकेशन जनरली लीड्स टू अ मोर रिलैक्स्ड chromatin structure, allowing easier access for transcription factors to DNA. Okay, now phosphorylation, it can contribute to chromatin remodeling as it is often associated with transcriptional activation. Okay, it phosphorylates and dephosphorylates the further downstream proteins. So phosphorylation is associated with transcriptional activation. Now coming to methylation, depending on the specific site of methylation, it can either promote or repress transcription. So all these three modifications can play a role in chromatin structure during transcription. The specific effect depends on the site of modification and cellular context. Okay. Moving on to next, which of the following is not a mechanism of epigenetic change? Which is not a mechanism of epigenetic change? Anyone knows the answer? Anyone? What is epigenetic change? What is epigenetic change? All of these are related to chromatin. Earlier we studied DNA methylation, histone acetylation, nucleosome repositioning. Only the alteration of a DNA base sequence in a promoter is different. So epigenetic changes involve modification to DNA structure that do not change the underlying DNA sequence itself. And it is talking about DNA sequence change. Okay, DNA base sequence alteration in a promoter. So this is incorrect statement. Okay, answer B. So epigenetic changes involve modifications to DNA structure that do not change the underlying DNA sequence. While DNA methylation, histone acetylation, and nucleosome repositioning, all are considered as epigenetic mechanisms that change the actual base sequence of DNA is not. Okay. So DNA methylation, it is addition of a methyl group to DNA and it does not change the sequence of DNA. So typically at cysteine bases, it adds a methyl group and often leads to gene silencing. Now histone modification, chemical changes to histone proteins that DNA wraps around affects the accessibility and gene expression. Nucleosome repositioning, the movement of nucleosome along the DNA strand alters the chromatin structure and gene expression. So all of these are associated with epigenetic changes except the one alteration of a DNA base sequence in a promoter. It could be another statement, any of the statement would come here related to the base sequence change or DNA sequence change. So it will be incorrect as epigenetic changes involve the modification to DNA structure but not to the DNA sequence. Okay, clear? Clear to all? Is it clear?
Chromosome repositioning.
Is it clear? Should I move ahead? Is it clear to all? What are epigenetic changes? So moving on to next question. Which of the following regions of histones undergoes chemical modification during transcription activation? Is it C terminus domain, central globular domain, N terminus tail domain, or all of the above? Region of histone that undergoes chemical modification during transcriptional activation. Histone modifications like acetylation and methylation. So they primarily occur at which of the following position? It is C terminal domain, central globular domain, N terminus tail domain, or all of the above. So histone modifications, which we already discussed, that it consists of acetylation and methylation. They primarily occur on the flexible N-terminal domain. Okay, N-terminal tail domain, which protrude from the nucleosome core. They significantly impact the chromatin structure and gene expression regulation. So the region where histone modification occur and undergoes the chemical modification during transcription activation, it is N terminus tail domain. Most of the modifications happens on this domain only. Modifications can loosen chromatin structure and they allow easier access for transcription factors to DNA, thereby activating transcription. And types of modification are acetylation, methylation, phosphorylation, and ubiquitination. Okay, we already discussed one of those is acetylation. Another is methylation. Methylation, phosphorylation, ubiquitination, simulation, ADP ribosylation. One is citrulation. Okay. So histones are subject to post-translational modification by enzymes primarily on their N-terminal tails, but also in their globular domains also. But, but most of the modifications occur in N-terminal only. Okay, like acetylation, methylation, ubiquitination. So these are different kinds of histone modifications.
Ma'am, I can't see anything on the screen, ma'am.
Screen is not visible.
Yes, ma'am.
Is it visible now?
No, ma'am.
No?
No.
Till when it is not visible? Earlier this question was visible.
Actually, my connection was lost, so I joined just now, ma'am.
Okay, okay. If something happens, please let me know. Okay.
Okay, ma'am.
Is it visible now?
No, ma'am. It's blank.
Not visible?
No, just I can see your profile picture. That's it, ma'am.
Not visible to anyone?
Yes, ma'am. It's visible.
Something wrong with you, Yakub?
Okay.
Visible to all.
Okay.
So we were discussing about the histone modifications. So most of the histone modifications occur at N-terminus tail domain, and we were talking about different kinds of histone modifications. So these are different kind of histone modifications. Okay. Coming to next, histone methylation is associated with which of the following? Euchromatin, heterochromatin, centromeric chromatin, or telomeric chromatin? histone methylation is associated with euchromatin, heterochromatin, centromeric chromatin or telomeric chromatin. Anyone? Anyone knows the answer? Histone methylation, which is a again a modification. So it is associated with euchromatin, heterochromatin, centromeric chromatin or telomeric chromatin. Which of the following is correct? So talking about modifications, histone modifications, one of which is histone acetylation. Histone acetylation. Acetylation, okay. It is basically associated with euchromatin structure, okay. Whereas histone methylation, it promotes heterochromatin remodeling, okay. Acetylation क्या करता है? It makes the histone group more negatively charged, which in turn disrupts its interaction with the DNA strand, essentially opening the strand for easier access. नेगेटिवली चार्ज कर देगा हिस्टोन ग्रुप को, which disrupts the interaction with the DNA strand, essentially opening the strand for easier access. This is the function of acetylation. Okay. Now, acetylation कहाँ पर होता है? It can occur on multiple lysine groups. Acetylation lysine groups पे होता है. Lysine residue of histone N-terminal tail and in different histones of same nucleosome which is thought to further increase DNA accessibility for transcription factor. Now talking about phosphorylation, by which euchromatin is regulated. This also tend to occur on N-terminal tail of histone. However, some sites of phosphorylation are also present in the core, as phosphorylation is controlled by kinases and phosphatases, which can add or remove the phosphate groups. So this can occur at serine, threonine, and tyrosine. Okay, present in the euchromatin. Since phosphate groups added to the structure will incorporate a negative charge again, it will promote the more relaxed open form again, similar to acetylation. In regards to functionality, histone phosphorylation is involved with gene expression, okay, DNA damage repair, and chromatin remodeling. Now, another method of regulation that incorporates a negative charge is ADP ribosylation. It can also add one or more ADP ribose ADP ribosylation. It can add one or more ADP ribose units to the histone and is involved in the DNA damage response pathway. Okay, so here histone methylation, here it is showing incorrect, it is B. Histone methylation is associated with heterochromatin. Okay, acetylation is associated with euchromatin, phosphorylation is associated with euchromatin, okay, but methylation is associated with heterochromatin. As histone methylation, it is an often a marker for condensed transcriptionally inactive chromatin, which is a characteristic of heterochromatin only. Specific methylation patterns can vary depending on the site and context. The overall trend is that histone methylation, it is linked to more compact or repressed chromatin state. Okay, euchromatin is associated with active gene transcription and associated with histone acetylation, while heterochromatin is associated with methylation and repressed gene expression. Is it clear?
Clear, ma'am.
Okay. H3 histone methylation occurs at which of these amino acid residue?
Option B.
Option B.
C, ma'am.
C.
Yes, ma'am.
H3 histone methylation. Sure. What about this? Arginine or serine? It is not serine, okay? It is arginine. Methylation of both H3 and H4. Both tails can occur on both arginine and lysine and not on serine, okay? It plays a critical role in transcriptional regulation. Most common site is lysine, where it is frequently methylated site on histone, but primarily these happen on lysine and arginine within the histone protein, particularly on the tail region of the histone H3. Okay. Methylation pattern on histone H3 can influence the gene expression with different methylation sites associated with either active or repressed transcription. But the sites are lysine and arginine. Now H4 acetylation. Anyone? Anyone knows the answer? H4 acetylation. It is incorrect. It is C. It is not A. H3 methylation is at lysine and arginine, but H4 acetylation is at lysine and serine. Okay. H4. Anyone is saying something?
So that's the difference between methylation and acetylation, ma'am.
Methylation and acetylation. We have discussed that acetylation occurs at euchromatin. It is mainly related to the creating the negative charge on the euchromatin, and it makes it more open, and it provides a methyl group. Okay, this provides a methyl group, and this acetylates it. What is the difference between acetylation and methylation, first of all? Major difference is euchromatin and heterochromatin, but what is acetylation and methylation? ये एसिटाइल ग्रुप ऐड करेगा, लाइसिन पे ऐड करेगा ये. ओके, लाइसिन पे. मिथाइल ग्रुप ऐड होगा साइटोसिन पे. फॉस्फोरिलेशन होगा सीरिन थ्रियोनिन टायरोसिन पे. एसिटाइलेशन क्या करता है? इट यूजुअली इंक्रीजेज द जीन एक्सप्रेशन, वाइल मिथाइलेशन, क्योंकि ये हेट्रोक्रोमेटिन पे है, हेट्रोक्रोमेटिन इज ऑलरेडी ट्रांसक्रिप्शनली इनएक्टिव, इट could suppress or elevate the gene expression depending on which residue is methylated. All the methyl group is added to cytosine residue only, and DNA methylation usually suppresses the transcription of a gene as it involves the heterochromatin, and acetylation usually increases the gene expression. Okay, so here histone methylation occurs at lysine and arginine, as it is talking about H3 histone methylation. So it occurs at lysine and arginine. And H4 acetylation, when we talk about acetylation, it occurs at lysine and serine. Where lysine is again major primary amino acid that gets acetylated in histone. Also, acetylation sites of H4 histone is also known, which is lysine 5, 8, 12, and 16. Okay, so at these particular sites of lysine, H4, on these particular lysine residues that are present on H4, acetylation occurs. Okay. Now phosphorylation.
D, ma'am.
Phosphorylation D. Very good. It is serine threonine. Phosphorylation is always related to serine, threonine, and tyrosine. Okay? No confusion in this. Where are DNA's 1 hypersensitivity sites found? Promoter and other regulatory sequences, intron sequences, exon intron boundaries, or 3' end of the gene. DNA's 1 की हाइपर सेंसिटिव साइट्स कहाँ पर प्रेजेंट होती हैं? Anyone? DNA's 1 हाइपर सेंसिटिविटी साइट्स होती हैं जो हमारी क्रोमेटिन में प्रेजेंट होती हैं, दैट आर सेंसिटिव टू क्लीवेज बाय DNA's 1. Okay. In this specific region of genome, chromatin has lost its condensed structure, exposing the DNA and making it accessible. अभी हमने पीछे पढ़ा था that euchromatin is highly sensitive to DNase 1 as it is loosely packed and open and makes access of DNA to DNase 1. So in the genome, in the specific region of genome where chromatin has lost its condensed structure, exposing the DNA and make it accessible, this raises the availability of DNA to degradation by enzyme, that is DNase 1. So these accessible chromatin zones that are functionally related to transcriptional activity, since this remodeled state is necessary for the binding of proteins such as transcription factor. अब ये जो accessible chromatin zones है जहाँ पर हमारा ये डिग्रेडेशन हो रहा है DNase 1 का, वो ट्रांसक्रिप्शनली एक्टिव हैं सिंस दिस रीमॉडल्ड स्टेट इज़ नेसेसरी फॉर बाइंडिंग ऑफ प्रोटीन्स, यहाँ पर हमारे ट्रांसक्रिप्शन फैक्टर्स आके बाइंड करेंगे, बाइंडिंग ऑफ प्रोटीन्स सच एज ट्रांसक्रिप्शन फैक्टर्स. अब ट्रांसक्रिप्शन फैक्टर्स कहाँ बाइंड करते हैं? इज इट इंट्रॉन, exon intron, 3' end of the gene, or promoter or other regulatory sequence? So these reasons have been shown to map many types of cis regulatory elements, including promoters, enhancers, insulators, silencers, and locus control regions. Okay, so here option A is correct. So DNA's one hypersensitivity sites are found in promoter and other regulatory sequences where transcription factors could bind to the promoter. So is it clear? Clear to all? Moving on to next question. Which of the following is not involved in chromatin remodeling? Histone acetyl transferases, NARF, SWI SNF, or DNA methyl transferase? Which of the following is not involved in chromatin remodeling? Anyone? It is DNA methyl transferase. Out of these, DNA methyl transferase is not involved in chromatin remodeling. Chromatin remodeling is the process of changing the way DNA is packaged in chromatin. This can involve moving, ejecting, or restructuring nucleosomes, where chromatin modifying enzymes play the role. These enzymes add or remove post translational modification from chromatin proteins, one of which is SWI SNF, a chromatin remodeling complex that uses ATP to destabilize histone DNA interaction. Okay, this SWI SNF, it is a chromatin remodeling complex. It uses ATP to destabilize histone DNA interaction. Now histone acetyl transferases, this these enzymes transfer an acetyl group to histone proteins and increases the gene expression. So it is asking which of the following is not involved in chromatin remodeling. So this is involved, this is involved. Now talking about NURF. It is nucleosome remodeling factor. Okay. It is a protein complex that plays a crucial role in altering chromatin structure by sliding nucleosome along the DNA strand, thus regulating gene expression. Similar to the SWSNF, it is also a protein complex. It also alters chromatin structure by sliding nucleosomes along the DNA strand, thus regulating gene expression by making genes more accessible to transcriptional factor. Okay, it is nucleosome remodeling factor. So everything is involved in chromatin remodeling except DNA methyl transferase. Also, other chromatin remodeling mechanism which are not in this options. So deacetylases, phosphorylation, methylation, phosphorylation, ubiquitination, sumolation, these are different mechanisms. These are different chromatin remodeling mechanisms. While IS ISWI, it is another kind of remodeling complex like NURF, SWI, SNF. Okay, so ISWI is another remodeling complex. It is imitation complex, imitation switch. CHD. CHD is another chromodomain helicase binding DNA binding. Okay, it is chromodomain helicase DNA binding domain. So it is another chromatin remodeling complex. Another is INO 80. It is inositol requiring 80. So these are different chromatin remodeling complex. Okay, and these are different chromatin remodeling mechanisms. Okay, so any of these could come in the question, so you have to note that which of the following is not involved in the chromatin remodeling. Okay, clear to all? Moving on to next, enzyme complexes that actively move or displace nucleosomes, hydrolyzing ATP in the process and causing transcription activation. Enzyme complexes that actively move or displace nucleosomes, hydrolyzing ATP in the process and causing transcription activation. I hope it is clear now. You should answer this question. I already explained in last question. Now you should answer. Enzyme complexes that actively move or displace nucleosomes, hydrolyzing ATP in the process and causing transcription activation. First of all, which of these is enzyme complex? Anyone? Anyone?
Option A?
Option A, 1 and 2.
Yeah.
Is it 1 and 2? From here you have to select.
Yeah. Yeah.
A.
Yeah.
Is it complex? Enzyme complex that actively move or displace nucleosome? Acetyl transferase acetylates or either transfer the acetyl group from one to another histone and it will transfer the methyl group. I have already explained the function of these two enzyme complexes, which is this. In the previous question, I have explained the function of these enzyme complex. Both, both of these have the same function that these enzyme complexes actively move or displace nucleosome, hydrolyzing ATP and causing transcription activation. So the correct answer is B, not A. Why A? Why you have chosen A?
Got confused, ma'am.
I already explained that these are two enzyme complexes which are involved in the chromatin remodeling. These are other remodeling complexes also, chromatin remodeling complexes which could be involved. Okay, these are mechanisms of chromatin remodeling. Okay, now if we talk about enzyme complexes, so either of these or either of these, any of these five could be there.
Okay, now I get it, ma'am.
Okay. moving on to next question. Heavily methylated DNA is associated with activation, repression, hyperactivation. Which of these? Of transcription in vertebrates and plants. Heavily methylated DNA is associated with dash of transcription in vertebrates and plants. Anyone? Anyone knows the answer?
Option A.
Activation.
Option C.
Hyperactivation. मिथाइलेशन क्या करता है? What is the role of methylation? पीछे एक क्वेश्चन में हमने डिस्कस किया था, आई होप हेयर, एसिटाइलेशन, मिथाइलेशन, फॉस्फोलेशन, इसका पॉजिटिव रेगुलेशन था, इसका भी पॉजिटिव था, बट इसका नेगेटिव रेगुलेशन था. लाइक इट एनहेंसेस जीन एक्सप्रेशन, इट ऑल्सो एनहेंसेस, बट मिथाइलेशन रिप्रेसेस द जीन एक्सप्रेशन. तो अगर हमारा डीएनए हैवी मिथाइलेटेड है, so it will be associated with repression of transcription and not activation. Okay. Heavily methylated DNA is associated with repression of transcription, as DNA methylation is a process which involves silencing of genes by preventing transcription factors from accessing the DNA. Therefore, heavily methylated DNA is linked to repression of gene expression. When a gene region is heavily methylated, it is usually not transcribed. So how could it lead to activation? It will definitely be repression. Okay, DNA methylation also plays crucial role in various biological processes including development, cell differentiation and genomic stability by regulating gene expression through repression always. Okay. So DNA methylation कहीं भी आता है, it means repression. Heavily methylated DNA means repression. Clear? Clear to all?
Yes, ma'am.
Coming to next, one of the epigenetic mechanism of chromatin inactivation. Epigenetic mechanism of chromatin inactivation is histone methylation, acetylation, phosphorylation or methylation. क्रोमेटिन इनएक्टिवेशन कैसे होगा?
डी डी मैम।
डी, बी और डी? क्या बोल रहे हो? रिपीट प्लीज.
डी मैम, आपने कहा था ना प्रीवियस स्लाइड में।
Yes, yes, correct. Very good. DNA methylation. It leads to chromatin inactivation. DNA methylation एक प्राइमरी एपिजेनेटिक मैकेनिज्म है दैट लीड्स टू क्रोमेटिन इनएक्टिवेशन बाय एडिंग मिथाइल ग्रुप्स टू डीएनए. टिपिकली जीन एक्सप्रेशन की साइलेंसिंग होगी इन द अफेक्टेड रीजन. वाइल हिस्टोन मॉडिफिकेशंस लाइक मिथाइलेशन, एक हिस्टोन मिथाइलेशन भी है, सो डोंट गेट कंफ्यूज्ड विद द डीएनए मिथाइलेशन. ओके. Histone modifications like methylation and acetylation are also epigenetic mechanisms, while DNA methylation is considered the prominent in chromatin inactivation. Histone methylation जो है it can either activate or repress depending on the specific site of methylation. Same with acetylation, it usually associated with open chromatin and it is associated with transcriptional activation. Okay. And phosphorylation is also involved in chromatin remodeling and also it is activation. Okay. So DNA methylation is inactivation or repression. Which of the enzymes is associated with gene silencing of activated genes? Which of these enzymes is associated with gene silencing of activated genes? अब यहाँ कंफ्यूज नहीं होना हिस्टोन मिथाइल ट्रांसफरेज से कि मिथाइलेशन है, मिथाइल ट्रांसफरेज ही करेगा जीन साइलेंसिंग. ठीक है, इट इज़ नॉट नेसेसरी. सो कौन सा एंजाइम है विच ऑफ दीज इज एसोसिएटेड विद जीन साइलेंसिंग ऑफ एक्टिवेटेड जीन्स? एनीवन? अब
सी होगा।
हाँ जी,
सी।
सी, डी एसिटाइलेज. यस, करेक्ट. कैन यू एक्सप्लेन दिस?
ये एसिटाइल ग्रुप को रिमूव करने से जीन साइलेंस होगा.
ओके. सो अर्लियर वी वर टॉकिंग अबाउट डीएनए मिथाइलेशन. डीएनए
हाँ.
मिथाइलेशन जो है वो जीन साइलेंसिंग से होता है, बट हिस्टोन की बात करें तो इट विल बी रिलेटेड टू डी एसिटाइलेशन. ओके. whenever histone deacetylase, it removes the acetyl groups from histone proteins. It leads to more compact chromatin structure and it makes it harder for transcription factors to access the DNA and thus gene expression is silenced. Okay. तो अगर हम मिथाइल ट्रांसफरेज की बात करें तो इट कैन ऑल्सो बी इन्वॉल्व इन जीन साइलेंसिंग डिपेंडिंग ऑन द साइट स्पेसिफिक साइट ऑफ मिथाइलेशन, बट इट इज़ नॉट द प्राइमरी एंजाइम associated with silencing the activated genes. It is talking about silencing the activated genes. Earlier we were talking about DNA where chromatin inactivation was involved. So chromatin inactivation में DNA methylation का रोल होगा, but gene silencing में histone deacetylase का रोल होगा. So it deacetylates, okay, removes the acetyl groups from histone proteins and makes it more compact chromatin structure. Okay, what does histone phosphotransferase do? Phosphotransferase क्या करेगा? फॉस्फेट ग्रुप को करेगा, बट इट इज़ नॉट जनरली स्टडीड इन रिलेशन टू जीन साइलेंसिंग, क्योंकि फॉस्फोलेशन phosphotransferase either will be involved in phosphorylation or dephosphorylation and not with the gene silencing. So here the correct answer is deacetylase. And talking about acetyl transferase, it also adds acetyl group to histone and it is associated with gene activation and not silencing. So the correct option is deacetylase. Methylation imprint of the genes is established in the dash germline. जीनोमिक इम्प्रिंटिंग पढ़िए. So the correct answer here is both maternal and paternal. Okay. Methylation imprint जो होता है germ line में, it is established in both the germ lines, both paternal and maternal. Okay. So DNA methylation is irrespective of the gender. As it is an essential epigenetic mark crucial for normal mammalian development. Moving on to next. Suppose female mice homozygous for the A allele of the IGF2 gene are crossed to male mice homozygous for B allele of this gene. Which of these two alleles will be expressed in F1 progeny?
Both A and B.
Both A and B.
Option C, ma'am.
Option C.
Yes, ma'am. Option C. Yes, ma'am.
it is saying that female mice homozygous for the A allele of the IGF2 gene are crossed to male mice homozygous for B allele of this gene. Which of these two alleles will be expressed in the F1 progeny? दोनों homozygous है, A, इधर भी homozygous है, B. So it will be both A and B. Correct. It is showing the incorrect answer, but it is C. So chromatin contains which of these?
Option 4.
DNA, RNA and histones.
Option 3, ma'am.
No, it's DNA, RNA and histones. RNA is also there in chromatin.
Okay, ma'am.
Okay. So here is the concept. The thread-like structure is chromatin. Nucleosome constitute the repeating unit of structure in the nucleus called chromatin. Chromatin condenses into chromosomes during cell division. Chromosome is a highly condensed form of chromatin. Chromatin present in two forms. Euchromatin loosely packed and stains light. Heterochromatin densely packed stains dark. Negatively charged DNA is wrapped around the positively charged histone octamer to form a nucleosome in chromatin, which is seen as beads on string structure when viewed under an electron microscope. So chromatin is made up of DNA, RNA and protein, which is histone. Chemical composition is this: DNA 31%, RNA 2 to 5%, histone proteins, which are basic proteins, these are 36%, and non-histone proteins, acidic proteins, which is 28%. Okay, so 20 to 30% part of histone is made up of arginine and lysine amino acids. Clear?
Yes, ma'am.
Okay. Additional information: DNA is the nucleic acid found in living system. DNA is the genetic material, came from Hershey and Chase. Proteins are polymers of amino acids, positively charged. Basic proteins known as histones. RNA क्या है? First genetic material थी. In some viruses, RNA is the genetic material. Okay. So here now we talk about few minutes or more talking about SMC complexes, which we will cover in the next class. Today we talked about chromatin remodeling and structure. So few questions related to SMC proteins also we will cover in next 10 minutes. So DNA filaments that develop inside the nucleus during cell division. Which of these DNA filaments develop inside the nucleus during cell division? Anyone? Anyone?
B, ma'am.
Chromosomes. Chromosomes are thread-like structures that develop inside the nucleus during cell division. These are made of DNA and proteins. So it is correct that chromosome is a DNA structure that resembles a thread and carries genetic information in the form of genes. Okay. Which of the following does not belong to significant chromatin protein class? Which of the following does not belong to a significant chromatin protein class? It is cohesins. SMC family of proteins include cohesions. Okay. Cohesins are important proteins which are involved in chromosome structure. They are considered a subclass within the SMC family protein and they are not a separate significant chromatin protein class. While histones, topoisomerase, and SMC proteins all are recognized as major chromatin protein classes. They are part of SMC protein complex. Their primary function is to hold sister chromatids together during DNA replication until they are separated in anaphase. And mutations in these cohesin subunits can lead to chromosome segregation errors also. So, talking about these, which of these does not belong to significant chromatin protein class? It will be cohesins. कोहेसिन्स जो हैं वो क्रोमेटिन प्रोटीन्स नहीं हैं, बाकी सब क्रोमेटिन प्रोटीन्स हैं. ओके. Which of the following plays a significant role in initial joining of sister chromatids after replication?
Cohesive ma'am.
Yes. Sister chromatids are bound together by cohesins during replication and they are kept together as chromosomes condense to metaphase and they are separated in anaphase. Which of the following is required for chromosomal condensation as cells enter mitosis?
Histones ma'am.
It is again cohesin. Chromosomal condensation as cells enter mitosis. Okay. So here,
Ma'am.
yes,
Option C ma'am.
yes. Also, condenses also have role in chromosomal condensation as cells enter mitosis. So condenses can also be the correct answer here. While cohesins are crucial for holding sister chromatids together during mitosis, they're not mainly have role in the condensation. Okay. So here they are although critical and crucial right after the replication, but the major role of chromosomal condensation will be linked with the condensins. They actively participate in the process of chromosome condensation by compacting DNA into tightly coiled structures. Okay. Chromatin is made up of, this question is repeated thrice. DNA, RNA, and proteins. Which of the histones below binds to linker DNA?
A, ma'am.
Which of the histones below binds to linker DNA?
Question not showing, ma'am. DNA, RNA, and proteins, ma'am.
Next, which of the histones below binds to linker DNA?
H1, ma'am.
H1, correct. H1, often known as linker histone, it specifically binds to linker DNA that connects nucleosome cores within chromatin fibers. While H2A, H2B, H3, and H4 form the core of the nucleosome around which the DNA wraps. H1 is the linker histone responsible for binding to the linker DNA between nucleosomes. Okay, so it is H1. Two copies of H2A, H2B, H3, and H4, and H1 is the linker DNA. H1 is the linker histone responsible for binding to the linker DNA between nucleosome. Which of the following has a strong structure with beads on it? Easy question? No? Beads. किस पे होते हैं beads? ये तो बचपन से पढ़ते आ रहे हैं. Beads on string. Anyone? Correct answer is nucleosome. When DNA wraps around the histone proteins, it forms like beads on a thread. Okay. Nucleosomes are formed when DNA wraps around the histone proteins. So this whole structure is known as nucleosome. And when this is viewed under an electron microscope, nucleosomes appear as beads on a string. Each nucleosome is made of DNA segment wrapped around 8 histone proteins, which are known as histone octamer. So the structure of chromatin plays an important role in controlling gene expression and replication. The closed structure of nucleosomes is not very accessible to enzymes that perform replication, transcription, and DNA repair. So the structure of chromatin can be disrupted, making the DNA more accessible to replication and transcription factor. And structure of chromatin can also have long range effects on gene expression. So strong structure with beads on it will be nucleosomes. Which of the following histones has the most comparable sequence across eukaryotic species? Anyone? Which of the following histones has the most comparable sequence across eukaryotic species? it is histone H3. It has most comparable sequence across eukaryotic species. The amino acid sequences of H3 and H4 histones are nearly identical in all the eukaryotes. So remember this thing, H3 and H4 histones का जो sequence है वो nearly identical है in all the eukaryotic species. Histone H3 is one of the 5 main histones that make up chromatin in eukaryotic cells. It is involved in structure of nucleosomes which form the beads on a string structure. These are highly conserved across the eukaryotes. These are dynamically modified by many post-translational modifications. Histone variant, which is H3.3, it is one of the highly conserved protein across eukaryotic species. It can be deposited in chromatin, often progressively replacing canonical H3 in nucleosomes over time. Also, this plays a role in maintaining the transcriptional landscape of cells and on emergence of specialized chromatin assembly pathways. So, the eukaryotic chromosomes, which are made up of DNA coiled and condensed around an octamer of basic proteins called histones. So, where H3 and H4 histones are nearly identical in all eukaryotic species. Sister chromatid split at, this is easiest question, today's easiest question. We have already covered this in previous classes. Sister chromatid split at which of these phases? Prophase, anaphase, metaphase or telophase?
B, ma'am.
B, anaphase?
Yeah.
Yes. Each chromosomal pair is divided into two identical and independent chromosomes during anaphase. So here sister chromatid split. During anaphase, the centromere of each chromosome splits, causing the sister chromatids to separate and move towards opposite poles of the cell. Throughout which of these phases, cellular DNA is uncondensed? This is also easy.
C, ma'am.
Telophase. Uncondensed.
Interphase, ma'am.
Yes, interphase. DNA exists in loose, uncondensed form called chromatin, while in other phases like prophase, metaphase, anaphase, and telophase, which are part of mitosis, DNA condenses into visible chromosomes there. Chromosomes ही तो देखते हैं उसमें हम प्रोफेज मेटाफेज एनाफेज टीलोफेज में, तो वहाँ तो कंडेंस्ड है, अनकंडेंस्ड कहाँ है? इंटरफेज में. ओके, DNA is uncondensed and exists as chromatin. Prophase में they start to condense into chromosome. Anaphase or telophase में they remain condensed during these phases also. Okay, the DNA of the cell is not compacted during interphase. Who coined the term chromosome? Who coined the term chromosome? It is Walder. Okay, this is memory based question, so we should not waste time on this. Color blindness is caused by mutation in the which chromosome gene in males?
B, ma'am.
X chromosome, correct. Gene which causes color blindness, inherited red and green types, is only on the X chromosome. So for a male to be color blind, the color blindness gene only has to appear on his X chromosome. Okay, as it is caused due to recessive genes. which occur on X chromosome. Who first presented the chromosome theory of inheritance?
None of them.
Chromosomal theory of inheritance was proposed by
See, ma'am.
first presented chromosomal theory of inheritance was first given by Boveri and Sutton. Okay, Boveri. Actually, it is Boveri, Correns, and Sutton. So, Boveri and Sutton, you might have heard this. Okay, so it is given by Boveri and Sutton in early 1900s. It is the fundamental theory of genetics. According to this theory, genes are units of heredity and are found in the chromosomes. Chromosomal theory of inheritance came into existence long after Mendelian genetics. As the time passed, scientists Weiss, Correns, and Schermak, they discovered chromosomes which existed inside the nucleus. Then Sutton and Boveri, they discovered and observed the behavior of chromosomes when the cells were divided. Okay, so here who first presented it will be Boveri and Sutton, and then later Correns gave his concept. The sex linked sickness is referred to as which of the following?
Option B.
Yes, it is color blindness. It is recessive disorder on X chromosome.
सीड्स थोड़े से बचे हैं और हाफ एम एस की 4 प्लेट बची है, या तो अगर ज़्यादा हाफ एम एस लगाना है,
4 बचे हैं सीड मतलब
4 प्लेट हाफ एम एस की और
हाँ।
सीड
सीड तो बचे हैं ना।
हाँ सीड तो काफ़ी
कल
है।
फ्रेश प्लेट बना के करते हैं, और मैं
ठीक
सोच रही हूँ उसको उसमें करेंगे हीट वगैरह में भी,
अच्छा,
तो उसमें तो एमएससी चाहिए हमें।
तो लेकिन हीट में ज़्यादा सा सीड भी चाहिए, हीट का तो वैसे लगाते हैं ना
सीड तो है, 4-5 प्लेट वैसे कर देंगे सिंगल लाइन एक्सप्रेशन के लिए, बाकी हीट वाला कर देंगे।
ओके
ओके,
जाऊँ मैं?
हाँ।
सीड
प्लाज्मिड क्या रहा?
प्लाज्मिड निकल गया है।
फाइनल कहाँ पड़ा अभी तुम्हारे स्टैंड, तुम्हारे को -20
निकाल के हम कहाँ रखें बताइए?
वैसे तो
अरे
मेरा
उसका
मन है
तो
लेट हो गया 10 बजे तक
उसका
पहुंचना
पैलेट
मुझे।
डिजॉल्व नहीं हो रहा, वो बहुत गाढ़ा हो गया, और पानी मिला दूँ
अच्छा,
जो मैनुअली निकाला था।
टोपो वाले है ना मेन तो, और बाकी वो वाले
वो
आया है
वाले-
प्लाज्मिड,
प्लाज्मिड किस में?
चेक नहीं किया होगा ना अभी तो।
वो कॉलम वाला नहीं, तो कॉलम इसलिए फेंका भी नहीं है।
अच्छा, तो कल सुबह फिर चेक कर लेना, कल ही
Okay.
लगाती हूँ फिर मैं, अभी तो नहीं।
तो प्लाज्मिड में अपने पास ही रखना।
हाँ ठीक है, ओके। There's some issue with the screen sharing. Next questions then we will cover in the next class, okay?
Okay, ma'am. Thank you, ma'am.
Okay, hope you learned something from me.
Yes, ma'am.
Okay.
Thank you, ma'am.
All the best. Thank you for joining.
Thank you so much, ma'am.